Today, Dr. Jo Randall will be telling us about her research here at CDU on black jewfish. So Jo, apart from being an amazing office mate, she's also an expert in marine biology, and she did her PhD at the University of Tasmania on, I can't remember exactly on what, but maybe you'll tell us. Yeah, so your postdoc here is between CDU and AIMS. And so it's on black jewfish, and you'll be telling us about the drivers of productivity in a changing environment. Thank you, Jo.
Thanks. Thanks for coming, everyone. This is going to be relatively short, so you can all start enjoying your weekend nice and early. As Clem mentioned, I've got a background in coastal ecology. I did my study in Tasmania. My honours was on southern bluefin tuna movements, and then I moved on to macroalgal forests for my PhD. So for the last 3 years, I've returned to my true research love, which is fisheries research. And I've been lucky enough to be in some really awesome environments and catch some really awesome fish and manage not to get eaten so far. Can I use that one? No. So a bit of background. Most people up here would know the black jewfish. It's an awesome fish. It's great to catch. It's great to eat. The swim bladder market has made it a really valuable fish. They're really open to black market fishing. They're really highly aggregative, so they're easy to target. So they're really vulnerable to overexploitation. So they need really robust management. All the sectors want sustainable fishery well into the future. They're the key focus of the coastal line fishery, and they're also a large part of the catch for Indigenous and recreational sectors. As I said, they're really aggregative, so they aggregate in large numbers in known catch sites. And in 2016-17, the population numbers suddenly dropped. So it's based on catch per effort data, and the catch was just dropping. And so there was a lot of concern. They introduced management and quota. And then suddenly this was followed by 2 years of really high recruitment, which was great for the fish, but also highlighted a need for more information on the environmental drivers to better manage the fishery and know what was going on and what would happen. So this project was set up with NT Fisheries and AIMS and CDU, and our role was basically to investigate these environmental drivers. So freshwater discharge in the wet seasons up here in the tropics, and kind of what was really influencing the health of the populations. So basically we set up a study, and we had 4 different sites. So 2 Perrin Islands and Sampan Reef were the riverine outflow sites, so close to the freshwater outflow in the wet season. And then we had 2 sites, Caution Point and Mitchell Point, over at Tiwi Islands. And we sampled 3 times a year over 2 years to get the dry season, late build up, and then just at the end of the wet season. And we collected a whole load of physiological and environmental parameters. So we really wanted to get a hold of changes dependent on the sites that we had at different times of the year, and the health and body condition of the fish. So we were looking at food web tracking with the stable isotopes. We were doing a lot of aging with Odoliths increments, all the morphometric, so GSI, HSI, which is gonad and liver compared to body mass, and also body condition index. And then we were looking at instantaneous growth with the RNA-DNA ratios. We've had a PhD student looking at the parasite loads and compositions, which is getting some really interesting data, and also looking at on the reef sites, the microbial communities in the water. So trying to trace this to the source water upstream. And then we collected all the environmental parameters, so the dissolved oxygen, salinity, temperature, and all the climate indices stuff. So we got a total of 220 fish, and then we also got an extra 160 different samples, so gonads and otolith and morphometric data off both fisheries and commercial fishes. In terms of results, we've got prelim results in now. Got a really good length-weight relationship. It does differ by site. So there is differences. It may reflect different stock structure at the different sites, but for the most part, a pretty good correlation there between them all. On the left there, we've got the Fulton's condition index. So that's basically just a relationship between length and body mass. And it's a little bit variable in the first year. If you remember, that was the 2019-2020 wet season where we didn't really get a wet season. In the second year, it's a lot clearer to see that for the most part, there's a reduction in body condition in the wet season. There's a bit of an anomaly there in the Parent Island site, so that's one of the freshwater outflow sites. A few different other indices there. We've got the hepatosomatic index, so that's the liver to body mass. Again, it's lower condition generally for the most part in the wet season. Again, the Parent Island site is showing a bit of difference there. On the bottom there, we've got water content, so that's basically the opposite, proxy for the opposite of the lipid content of the tissue. So for muscle tissue here, when we see high water content, that's a lower lipid content, so worse condition. So you're seeing again that higher trend for higher water content in the wet season. The RNA DNA ratios, on the right there, again, these are lower generally in the wet season. Again, the Parent Islands is a bit of an anomaly there, so it would be interesting to figure out what exactly is going on at that site. Onto reproduction. We did a lot of work, got a lot of extra samples for reproductive work. But first of all, we had to kind of figure out what we were doing, and we wanted to make sure we were consistent and really on board with what NT Fisheries were doing and all the other researchers. And it became apparent that everybody wanted a simplified staging guide. There was about 7 or 8 different reproductive stages originally, and some of them you needed a microscope, so it wasn't very helpful out on a boat. And what we really wanted to know was basically, were they juvenile, were they reproductively mature, were they spawning, or were they spent? So we kind of made this up, and it evolved into something that we thought we'd make available to everyone. So we've made that available to commercial fishers as well, because they like to give a bit of an eye on what the fisheries guys are actually doing on their boats. So everybody's quite happy with that one. So for this gonad work, we had pretty much 400 fish, and traditionally, it's thought that the spawning season is late in the build-up, so October, November. And the interesting thing we're finding is that, particularly with the males, we're finding that well on into the next early year as well. So whether that's a serial spawning when the conditions are right, or whether they just have an extended spawning season to what we originally thought, it would be good to have a hell of a lot more data to try and tease some of that out. On the top graphs there, that's males and females, and that's just the GSI, so the weight of the gonad to the body mass. You'd expect that to be highest when they're actually at spawning stage. So you see that September, October, November is a peak for both males and females. And then we've just got F2, so that's mature, and then getting ready to spawn and spawning stage down the bottom with the M4 and F4. And you can see, again, there's that reproductively ready to spawn rate at that October, November time. Odoliths were a real challenge with the jewfish. I don't think anybody's had an easy time with NT jewfish with the aging. Some of them are beautiful and some of them just give you a headache. We've tried a few different methods. We've tried the cut and burn, which is the golden there, where you cut them in half and you blowtorch them. It was about as successful as the slow speed saw method. So we kind of left that behind. What we found was that if we just used the slowest speed possible and the sharpest blades possible, it took quite a long time to get through an entire otolith, but we were getting the best results with that. In the end, we were getting NT Fisheries plus CDU to age them, and we were still getting regularly 1 year difference. And even though that doesn't sound like a lot in a fish where they're generally below 6, 1 year can make a big difference. So we've ended up sending them to the fish aging services, and we're developing a protocol with them and get some annotated reference slides together, and we'll go again from there. We got some really interesting stuff out of the stable isotope food web tracking work. We looked at carbon, nitrogen and sulfur, and these look a little bit confusing, but basically what we're interested in is those light blue clumps in the triangles in each of the 3 plots. They're actually the jewfish. So they're incredibly tightly clumped in there, which is indicating that they've got a really constrained preferred diet, which they'll kind of consider the chickens of the sea. Like, they will eat anything and everything, but I think this is kind of saying that they do have a preferred diet that they're sticking to. So probably something that's high energy and easier for them to catch. On the yellow is, we did a whole heap of sampling from floodplain vegetation. So in the wet season, anything that was likely to end up coming down the river into the food chain. And then the circles on the left are freshwater. So green is the producers, and the darker blue there is the consumers. And then on the right, the triangle is the same with the marine. And you can see that for all of the isotopes, the carbon, nitrogen and sulfur, it's pretty much dead centered on marine. So there's not really any freshwater influence in the diet at all, which is interesting because they're not really taking advantage of that massive freshwater influx of nutrients. This is just pulling it apart a little bit. So we've got dry season and wet season. It's all the same data. It's just separating. You can see it doesn't matter if it's dry season or wet season. They're still really focused in that marine side. And then we have some more, and it's just separating it out even further. So on the left, we've got the freshwater producers at the top and the consumers, and you can see there's really no overlap in those clumping of the jewfish there. And on the right is the marine. So the green there is, on the top is the phytoplankton, and yellow is seaweed. And then in the bottom there, you've got the crustaceans in green, and the yellow is small fish. So it looks like they're pretty centered over small fish, or probably more so the crustaceans. Working on getting some squid samples at the moment. We weren't able to get any of those, so we'll chuck them in as well. It's quite hard to get marine consumer samples because the jewfish with the barotrauma invert their stomachs immediately, so we weren't really able to collect a hell of a lot. I mentioned that there's a PhD student looking at the parasite loads and the species composition. I don't really know a lot about this part of the project, but she's given me a slide here with, on the top is the Perrin Island, so that's one of the freshwater sites, and on the bottom we have Caution Point, which is up on Tiwi Islands. You can see on the left the environmental parameters are much more variable, close to the freshwater outflow. So you've got those dips and peaks in temperature and salinity, which are the grey and the blue there, pink there, sorry, at the top. Compared to Caution Point, there's much less variation there. In terms of parasites, so Perrin Islands, we've got the yellow in the middle, that's Oriento. I'm not even going to try and pronounce it. What we weren't expecting there is they're showing a peak in that parasite, which is a common parasite, in the middle of the dry season. You'd expect with parasites of this kind that you'd be getting a peak in the warmer waters, but it may be that as they have an intermediary host, maybe there's a lag time, but it's still really early days with the parasite work. On the monogenia, there's that peak. I think they're just a gill parasite. So again, they're showing the mid-dry season peak as well. And this is pretty different to caution point, where you wouldn't expect so much fluctuation in the environment. But here we're getting the expected peak we would expect in the freshwater sites in the late wet and the build-up. So there's some interesting results there, and hopefully we'll find out some more in the coming months. Miriam's been doing a lot of work on the water source tracking, so using DNA from samples at our sink sites, so our aggregation fishing sites, and then comparing that with samples we're taking up in the freshwater source, potential source sites. She's using Bayesian mixing to look at the bacterial gene tags to be able to match the input from the freshwater sites into these sink sites. What we're finding is in the first year where we've run the study, we're not really getting a freshwater signal at any of the sites at all. We did have a really small low wet season in that first year though, so we decided to run it again last year in the wet that was much more substantial. And we've done a lot more sampling during the tide cycle, so instead of just concentrating on getting the outflow at the freshwater sites, we're also getting samples at all stages of the tide cycle from the aggregation sites. So the data's not in yet, but it should be in the next couple of weeks. I think this work, regardless of the findings, it's going to be really useful as kind of a backup and another source of information on why we're getting some of the other results, particularly the food web tracking. I wasn't sure that was going to work. This was the really expensive ARIS system that we really wanted to use this as fisheries independent means of doing abundance and stock structure. So it was really an assessment of how useful it would be in a shallow coastal environment. We used it on sampan, so that's our really shallow site. Like I think it was up to about 5 metres when we were out there, and this was a really calm day at that site. So you can see it was a bit hairy. There was quite a big fear that it was going to get stuck on reef on the bottom, and I wasn't going to jump in to try and find it. We did get about 20 blocks of footage that's being processed at the moment. I mean, I guess the pros are it's got really excellent resolution. You could distinguish young gear. You don't have to use baits on it like the brubs. Doesn't matter if it's nighttime or how turbid the water is. But you can't mount it on the bottom of the boat. Any kind of movement is going to blur everything. And yeah, in the coastal areas, I'm not sure how useful it is going to be. It would be very easy to kind of get it stuck on the bottom and not be able to get it back up. But yeah, we're going through the footage now, so that's kind of pending. And upcoming work, we're going to look at compound-specific isotope work to try and further pinpoint that food web tracking. The Odolith work is ongoing. The water source tracking should be results pretty soon. The PhD parasite project still has a fair way to go. And we're hoping to have a new PhD student, if she's successful next year, looking at some skin microbiome and gut microbiome stuff that we collected along the way, which might tie in really, really well with the parasite work. And that's all I have for you guys today. Short and sweet as promised. Anyone have any questions?
We only have the one microphone today.
Okay.
Big stand outside here.
With your stable isotope stuff, what size classes did you get all of them? Because I'm just wondering.
Size classes of jewfish?
Yeah.
No, we had for the most part they were adults. We did have a number of juveniles, mostly from the one site. So we did have about 12 juveniles in there as well.
And they still panned out with purely marine as well? Okay, that was what I was interested in.
Yep, so all, regardless of site or year or season, they're all grouping in that one. Yeah.
Thanks, Joe. I know a bit about your project, but I think your results are really interesting because they're different to what a lot of people have found with other species of coastal marine fish, in that freshwater inflows to those systems don't seem to be benefiting black jewfish, whereas the productivity that's brought in from the big wet season flood seems to boost productivity and help a lot of fisheries. So I guess my question is, do you have it? What's your best guess as to why it's almost not it's a disbenefit? The flows seem to decrease condition, and that's what your results are showing, and they stay focused on marine food sources. Do you have any guesses after doing this project for a few years and saying them?
I mean, I can guess. One idea would be that they really hit the one food source. I believe it would be small crustaceans, and I've heard some anecdotal evidence that in the big wet season, the normal size wet season recently, for a while the numbers did drop, and the ones that were being caught by the commercials were spitting out squid rather than the usual crustacean. So it could be that they're moving offshore to chase the prey source that's also moving off, and then the stragglers that are left behind are eating a suboptimal food source. But that's, it's just conjecture at the moment.
So do you have some isotopic data from the bigger wet season, or what you showed us, was it basically that very poor wet season that we had in 2019?
Most of the freshwater isotope samples were from the first wet season, but a lot of the marine stuff and zooplankton and phytoplankton we resampled in the second as well.
And no difference.
Well, it's all on the plots. So yeah, the marine was marine, and the jewfish weren't moving. So the jewfish that we sampled and analysed were from both wet seasons as well. So we did get the jewfish in both wets. No other questions?
Great. Thanks very much, Joe. Great talk.